All right, hello everyone. This week we are going to do a lab on metabolism. And in the metabolism lab, we're going to examine different types of chemical reactions and we are going to take a look at how metabolism work in the uva lobata, which is a sea lettuce. Okay, so metabolism is the sum of all the chemical reactions that are occurring inside of a cell. And these chemical reactions are essential for life. Two examples of a chemical reaction that can be occurring is catabolism and also anabolism. So, catabolism is a type of chemical reaction where large organic molecules are broken down into smaller ones. As this process is occurring, energy is being released. So, an example of catabolism is you taking in bread. Bread is made up of carbohydrates. Now, in order for us to function, we need glucose. So, the carbohydrate that you consume from bread is broken down by the cells in your body into glucose, which is the molecule that can then be broken down into ATP. On the other hand, we have a type of chemical reaction called anabolism. Anabolism is sort of the opposite. Anabolism is using small molecules, combining them together to build large molecules. And this reaction requires energy, requires the input of energy. An example that you can think of is taking amino acids, joining them together to build proteins. And with that, there are four macromolecules that are essential to life. So these are the building block of life. Carbohydrates, it's a very large molecule. Carbohydrate is broken down into its simplest form, which is monosaccharides. So think of glucose. Lipids, broken down into its simplest form, is fatty acids. Proteins, broken down into its simplest form, is going to be amino acids. So most of these things we can obtain it from our diet. Nucleic acid is broken down into nucleotides. Our DNA are based on the nucleotides. All right. So there are other chemical reactions that can occur inside a cell as well. These chemical reactions, the respiration, is essentially breaking down glucose in order to get energy. Aerobic respiration is when a glucose molecule is broken down using oxygen. We get a lot of ATP this way. Anaerobic respiration is when glucose can still be broken down, but because no oxygen is inputted, there's going to be less ATP that are yielded. Larger organisms usually perform aerobic respiration so that they can get more energy from breaking down a single molecule of glucose. Other types of chemical reactions, this one right here is not occurring inside of human cells. Okay, it's not occurring inside humans because we do not have the ability to perform photosynthesis. However, photosynthesis can occur inside bacteria and plants because they have the chloroplast organelle which can perform photosynthesis. So essentially in photosynthesis, light energy is needed. So you take a look at the reactants, what needs to be inputted into photosynthesis. We have light, carbon dioxide, and water. So if you take a look at the cross section of the leaf, we have these tiny stoma, plural stomata. These are the tiny pores that you find primarily on the bottom of the leaf. This is where carbon dioxide enters. Okay? Water is going to enter in the root, and then when light energy comes in, it is going to hit the chloroplast. And the chloroplast is an organelle that contains a pigment called the chlorophyll. This is the pigment that actually absorbs the wavelength of light. And the wavelength of light that is absorbed is usually between 400 700 nanometer. Pretty much in most plants, all the wavelength of light can be absorbed except for the green wavelength. So because green is not absorbed, it's reflected back. That's why leaves are green. So with the photosynthesis reactions, hence plural, there are many reactions that are occurring inside photosynthesis. The first one is the light dependent reaction. In the light dependent reaction, what happens is a molecule of water gets split. So light comes in and it split up water. This is photolysis. And as a result of that, oxygen is produced. Now, when water is getting split up, it releases a little bit of energy. That energy from photolysis is going to be used to reduce carbon dioxide into glucose. So in the light dependent reaction, water is split and then we get oxygen as a result. When water is split, a little bit of energy is released. That energy is used to reduce carbon dioxide into glucose. Now, photosynthesis overall is a type of redox reaction. And in this redox reaction, it allows for another reaction to occur, which is called carbon fixation. Carbon fixation is just when we go from an inorganic carbon to an organic carbon. In this case, the carbon dioxide is the inorganic carbon, and then when we form glucose as a result, the carbon in glucose is considered organic. Now, we also have another type of chemical reaction called cellular respiration. So now plants have produced that glucose molecule. They are going to have to break up that glucose molecule in order to get energy. So if oxygen is available, they can break down that glucose molecule, and as a result, the products now are going to be carbon dioxide, water, and energy as a result from breaking down glucose using using oxygen. Now, cellular respiration is a type of oxidative reaction. In the oxidative reaction, this is when we are converting an organic molecule back into an inorganic molecule. So we're converting glucose back into carbon dioxide. If we take a look at the cellular respiration equation, there is something that we need to talk about before we finish up, and that is the RQ quotient. The RQ quotient is the ratio of, you can think of it carbon dioxide expired to oxygen consumed. You can also think of it as oxygen to carbon dioxide whenever we break down a certain molecule. So in this example, whenever we break down one molecule of glucose, this requires 6 oxygen molecule and we yield 6 carbon dioxide. Now, 6 to 6, that's a 1-to-1 ratio. Now, this only applies to when we break down glucose. If we break down other molecules such as sucrose, fructose, if we break down proteins, carbohydrates into their simpler molecule, it may not always be a 1-to-1 ratio, but the RQ quotient ranges anywhere between 0.7 to 1, depending on whatever composition of chemicals that we're breaking down. All right, so before we leave, let's take a look at the photosynthesis and cellular respiration again. Okay. Now, if you take a look at it, these two equations are essentially the opposite of each other. And what's occurring in photosynthesis is that we start off with carbon dioxide, water, and light. So these are the reactants, what is inputted, and the products are going to be glucose, oxygen, and water. In cellular respiration, we start with glucose and oxygen, which are essentially the product of photosynthesis, and that becomes the reactants of cellular respiration. And as a result, we make carbon dioxide, water, and energy. So these are the products of cellular respiration. Now, plants, they are performing both of these reactions simultaneously in the cell. And if you think about it, when there are more light available, such as daylight, the plants are going to perform more photosynthesis than cellular respiration. And in the nighttime, they're going to perform more cellular respiration compared to photosynthesis. But at the same time, these reactions are occurring simultaneously in the plants. Now, in today's experiment, we are going to look at metabolism, which is the sum of all chemical reactions in the ova lobata. And this species of sea algae sea lettuce is found within the southern coast of California. So, understanding their physiology will really help us understand how energy is transferred through different trophic levels. So, in this experiment, we are measuring the amount of dissolved oxygen. This is the oxygen that the plants, pretty much you can think of dissolved oxygen as leftover oxygen. So, when the ovalobata perform photosynthesis, they are going to produce oxygen. They're going to use some of that oxygen to break down glucose in order to get ATP. Any leftover oxygen that the plants do not use themselves are going to be available to consumers like ourselves. So, in this experiment, when you go to your lab bench station, the first thing you do is take a picture of the setup, because when you're done, you want to return everything how you saw it. And once you have taken a picture of the setup, go ahead and turn on the LabQuest machine. The button is going to be up here on the side. Turning on this machine in the beginning is necessary because sometimes it may take 1 or 2 minutes for the machine to boot. So just after you take your picture, turn on the machine. Then what you're going to do is have someone put in 200 milliliter of seawater into each jar. You're working with 3 jars. Each jar will get 200 milliliter of seawater. And the key word is seawater. If you put in any other type of water, it will kill the ovalobata. And then you'll notice that you have a clear jar, a blackened jar, and then you're going to have a jar with just seawater. This is going to be your control. In the clear jar, you're going to take 4 grams of ova and put it in. Now, it doesn't matter if you have 2 pieces of ova, 4 pieces of ova, 10 pieces of ova. It really doesn't matter, but the final mass of ova that goes into the clear jar needs to be 4 grams. And then you also need to add 4 grams of ova into the darkened jar as well. And then you're going to measure the starting water temperature with the thermometer in each jar. Do this before you start. And remember, the temperature has to be in degrees Celsius. All right, so once the 4 grams of ova are in the clear jar, the 4 grams are in the darkened jar, and the blank one should just have seawater, you're going to start measuring the dissolved oxygen. And the first measurement that you're going to take account of is going to be at time 0. So what you want to do is put the jar onto a centrifuge so that it can spin and so that you can get a clear dissolved oxygen reading. Now, there's only one centrifuge per group. So every time you do the reading, you need to put one jar on, open up the cork in the lid, put the dissolved probe in, and then you can measure the dissolved oxygen reading. When you're done with the clear jar, remove it out and then put your dark jar on, put the probe in, measure the dissolved oxygen, and then when you're done with that, take the dark jar off and then put the blank jar on. Again, take out the cork on the lid, put the probe in to measure dissolved oxygen. Now, the probe is connected to the LabQuest machine, and that is how you get the reading. The reading is going to appear on the LabQuest machine. Then you're going to set your timer for 10 minutes. The clear jar is going to be stored under the light. The black jar, you can put a bucket over it to keep it dark. And then the blank control, you also put it underneath the light as well. You're going to leave them in that condition for 10 minutes. After 10 minutes, you repeat the reading. You need to put the clear jar on the centrifuge when you're getting the reading. When you're done, remove the clear jar. Then put the dark jar on the centrifuge, get the reading, take the dark jar off, put the blank jar on, get the reading, and then remove the blank jar. Return these 3 jars back to where they need to be. So the clear jar goes back under the light, the dark jar, you may want to put a bucket over it, and then the blank jar goes back under the light as well. Set a timer for another 10 minutes and you're going to repeat this process for a 1 hour time frame. Okay, so as of right now, go ahead and work on the hypotheses. What do you think the oxygen level is going to be like between these groups within the 1 hour time frame. Now, as you are collecting your data, there should be one person in your group that is making an Excel sheet. So the Excel sheet will look something like this. Now, this is an Excel sheet that is not realistic because your data will not be as clear as this. Usually, what happens is that your data fluctuates a lot within the first 20 minutes. It's not until after the first 30 minutes where you start to see a trend. Okay, so overall, the ova that is in the clear jar, okay, because it's exposed to light, there should be an increase in dissolved oxygen concentration over time because it's able to perform photosynthesis, and with photosynthesis, it does produce more oxygen. The ova that is in the darkened jar, this one should be showing a decline in oxygen over time because it's getting less light, and as a result, it's going to resort to performing more cellular respiration than photosynthesis. So oxygen levels probably will drop within the 1 hour time. Now, the blank control jar, okay, its line is somewhat stable, but there is a slight increase. Think about why the blank jar may show a slight increase over time, and we can discuss this in class together. So, again, when you are doing your graph, make sure you have all three lines into one graph so you can see the comparison. Make sure you have your axis title and make sure you have the correct unit. Only one person in your group needs to do this. Okay? So when you're done, I will be checking pages 17 to 19 and I will look at the graph for your group and then you'll get a stat. Okay? All right, I will see you in class soon. Bye bye.